thank you very much for this kind introduction, and thank you for inviting me to come here. It's been 13 years since I was in Seattle at A&P, and it's really great to come in here. I should have come more often. Just a suggestion in case you didn't have any speakers next year. So I'm going to talk to you. So I'm by training a neuropathologist, anatomic pathologist, and molecular pathologist, and I do research mostly in epigenetics of brain tumors. But what I'm going to show you today is pretty much applicable across any solid tumor, and I hope you're going to see where I see the molecular pathology going forward. This is some required disclosure statements from your institutions, and I like to start sort of on an inspirational note, as I always run my lab meetings or my division meetings. Is why are we here? Why are we here today? And I always say, well, we're here to cure cancer, right? That's the single thing why we're here. And if we are not here to cure cancer, then we have no business of being here, right? And I'm going to tell you three stories, focusing on precision diagnostics and why I think it matters. Then we're going to talk to you about how we pathologists can contribute to the development of novel therapies and drug delivery, because we have a unique opportunity by seeing tissues, observing what's happening in there. I'm going to talk to you molecular monitoring, where I see molecular pathology going. But first I want to tell you a little story from when I was in the medical school, which is actually not as long ago as you might think. I had a fantastic mentor, fantastic teacher, and that was in the days when fish was hot. So I'm kind of dating myself, and he insisted that the worst career that a young person in medical school can choose is a pathology. Because he believed, and he predicted, that very soon we're going to have molecular techniques and machines that we're going to just throw the tumors in, they're going to spit out the mutations, and smart chemists are going to make the drugs matching those mutations, and we will not need those pathologists with their obsolete microscopes and H&E slides, because everything will be resolved with technology. So I obviously became a pathologist, which tells you two things. First of them is I don't listen to advice very well, which is what a lot of mentors of mine would agree. And the second thing is that even people who accurately predict the future and what's going to happen can be completely wrong in predicting how the future is going to be implemented. Because it is the time that we need pathologists more than ever before, exactly because we have those smart machines that are spitting out the tons of mutations and information about tumors, and that's what I'm going to try to show you today. So the idea of precision medicine, I'm sure it's familiar to all of you, is this idea that we'll go from histological pathology, so broadly annotates diagnosis of a tumor as a glioma, and ineffective therapy that maybe hits 1 out of 50 or 100 people. We're going to move to genomic or precision medicine. We're going to be able to batch patients into molecular characterized tumors to which the smart chemists are going to invent the molecules, and we're going to be able to target them more specifically. But as you all know, it hasn't really panned out completely. Mutations don't really always mean the same thing. So when we think about cancer, there are multiple alterations that are behind cancer. There's a mutation, this is in DNA. Then there's a change in expression, which we measure through RNA, and then of course proteins, right? But the one thing we're going to be specifically talking a lot today is DNA methylation, which is an epigenetic change, which is sort of like a font that changes the text of a DNA code. Over the last 7 years, we've built a state-of-the-art precision medicine in molecular pathology at NYU. We received the FDA approval for NYU Langone Genome Pack, which is currently the largest FDA-approved test for cancer. We're the first ones in the United States to launch a whole genome DNA methylation with machine learning for diagnosis of brain tumors, expanding now in multiple different cancers. And another one is an NYU Fusion Seeker, which is our comprehensive RNA NGS assay looking for known and unknown fusions in cancers, and many other things. So we have all these tools and everything what we are doing. But one of them we're going to be talking about is methylation. And why do we start working on methylation now several years ago as well? Because mutation present only part of the story about a tumor. And I call that a BRAF V600E paradox. It's a mutation that is present, every pathologist knows, every oncologist knows. It's a mutation that's present in many different cancers, but it has completely different implications in malignant melanoma, one of the most malignant tumors of the human body, or pilocytic astrocytoma. And I always say mutations are, to cancer and epigenetics, what is real estate to New York. It's the location that matters, right? Every mutation evolves on the background of the developmental changes and what is effectively epigenetics. So mutations don't tell the full story, And I'm going to show you the tumor mutation. The tumor behavior is highly context, which is basically cell of origin and tissue of origin development dependent. And we can do a better job classifying brain tumors and understand their molecular patterns. So we can better predict response to the therapy, we can predict outcomes, we can classify patients, identify high-risk patients, make better therapies, better clinical trials, and most importantly also, hopefully in the future, have better early diagnosis tests for prediction and follow-up. So when I was in neuropathology fellow, this is how we trained. This was all was needed. An H&E slide, and it was like a glioblastoma grade 4, and they were all the same. A medulloblastoma grade 4, and they're all the same. And it was a ependymoma, maybe grade 2, maybe grade 3. We could never agree on that, and it didn't really matter, right? So the times have changed significantly because now, based on many landscape studies, we don't define tumors histologically anymore, but molecularly. And we have noticed over the years that every one of those histologically the same looking tumors can be actually subdivided in multiple epigenetic subgroups. So here you see, for example, a subtype of a glioblastoma, now has a little bit of a different name. It's H3K27 mutated glioma, medulloblastoma, let's say sonic hedgehog type, and then Raleigh, which is now a C11orf95 fusion tumor. So precision for molecular diagnosis helps you to identify better characterization of the tumors and select better treatment options. This is an example of two patients with a medulloblastoma from NYU. You can see the red arrow like pointing to two big tumors in the posterior fossa, which on a traditional diagnostics both get the same diagnosis. But one of them ends up being a Wnt tumor with almost 100% chance of survival, and the other one ends up being a group 3 tumor with about 30% chance of survival. So it's hard to argue that these two tumors, although they look the same, are in fact the same disease if one child is almost certainly to survive and the other one has only 30% chance of surviving. So they have a different risk of recurrence and they should be managed differently. They need different clinical therapy management. I'm going to show you down the road different monitoring. So why is methylation so interesting? Well, DNA methylation is really a fingerprint. It's a memory that reflects lineage and cell development. Everyone in this room has started from a single cell. And that single cell proliferated, and all the cells share the same genetic information. And yet, the genes expressed in your heart and genes expressed in your brain are completely different. And that's all a result of a transcription, which is regulated by epigenetics, and one of the factors, DNA methylation. DNA methylation can also be a fingerprint, a signature of mutations that caused distinct epigenetic change. For example, IDH mutation is going to cause diffuse hypermethylation, K27M mutation is going to cause diffuse hypomethylation, and many different variations in between. DNA methylation is also very robust because it survives formalin fixation and paraffin embedding really well. It survives thousands of years. You probably saw the studies that you can do Neanderthal DNA methylation, right? Because if DNA survives, methylation will survive as well because it takes energy to remove those methyl groups. But if the site was methylated before the cell died, the DNA is going to remain methylated. So as long as DNA is stable, methylation will sit on it. It's also very simple computationally because every site can be either methylated or unmethylated. So it's a binary problem opposed to, let's say, expression, which can range from 0 to infinity, right? So there's no limit of how many transcript of RNA you can have per cell. It depends how high it is. And also DNA methylation does not degrade with time. The RNA expression will always vary based on how long was the tissue out of the body, was it like sitting around, how was it degrading, and RNA much more degradable. So you can start with sky-high levels of RNA. They're going to deteriorate really quickly because somebody left it over a heater, or you can start with extreme medium levels of the RNA expression. They're going to stay stable because the surgeon was really good and put it immediately in formalin and conserve whatever it was, and it depends on the age of a block and everything that's going to degrade RNA. So DNA methylation is very good because DNA is a more stable molecule, and DNA methylation just remains attached to it. This is published data, and you may have seen it by my fellow who was presenting here a few months ago, a month ago maybe, when she was interviewing for a talk. And it's the work that we started in about 2014 with Andreas von Daimling and Stefan Pfister in Germany. We got this idea, let's map out DNA methylation map of all the brain tumors, and let's design a classifier that for every DNA methylation, for every type of a brain tumor, we're going to find a DNA methylation signature. And you can see it's a little bit busy slide, but you can see here that on the left side you have all these color bars that indicate a specific type of a tumor in different colors, from embryonal to glioblastomas in green, glioneuronal in orange, and so on. And they run all this to DNA methylation array chip, and you see on the right side is a heat map, which is basically composition of the same shapes that you have in this chromosome-like diagonal that indicates the distribution of DNA methylation with the different colors. And you can see, you can zoom in on this top right angle that you can see a triangle of all the ependymoma. So within the ependymal cluster, you can also see all the different methylation subtypes of an ependymoma. So the new classifier version 12 now defines pretty much all of the 2021 WHO CNS entities and includes about 180 molecular classes and subclasses that we use. So we went from about 80 in the original 2018 Kapper paper to about 180 that we have now, and expanding beyond. So it's always good to show a great example that sort of highlights what is important of DNA methylation. This was one of our first patients that we did. It was a 17-year-old girl who presented with a seizure. You can see imaging showed like an enhancing tumor where the red arrow is, and it was she underwent subtotal resection with a pathology diagnosis of a malignant tumor. And you don't have to be a doctor, pathologist, an oncologist, and parents definitely don't like it when the diagnosis is malignant tumor because you really can't treat based on that. It was in a smaller institution, so her slides went to two very esteemed institutions in the country, and one called the glioblastoma, grade 4, and the tumor board recommended radiation and temozolomide chemotherapy. It went to another, more pediatric-focused hospital, and they said, no, this is what they called back then, it's a PNET. It's a primitive neuroectodermal tumor, grade 4, and they recommended craniospinal radiation and the PNET chemotherapy. So two very different diagnoses for a patient, for somebody who's young, who's going to live with consequences of the treatment for the rest of life. So as I always say, with a bit of a tongue-in-cheek, is what do you do when you have two discrepant diagnoses from two high-end institutions? Well, you come to NYU, right, for the third opinion. And it is a big problem, right? There's a reason why oncologists say if you send your slides to 3 pathologists, you're going to get at least 4 diagnoses, right? So she came to NYU for third opinion. We ran the DNA methylation, and we diagnosed the tumor as a pleomorphic xanthosarcoma with a WHO grade 2. So what are consequences of getting the diagnosis right or wrong? So first of all, we have the correct diagnosis. It was a PXA, and we actually did the BRAF testing. It was a BRV600 mutant, so it was a PXA. We gave her a grade 2. We recommended gross total resection. She underwent gross total resection now, it's going to be 6 years ago, and she never recurred. Finished college, I call every year there, ask her how is she doing, and they say, yeah, she's fine. She's just following up, and she never needed an additional piece of information, an additional piece of therapy. But what I would also like to emphasize, and I didn't put on the slide, is we also save tons of money because radiation costs a lot of money. So we show this case to all the insurance payers that don't want to pay for methylation. We say for a couple of $100, radiation costs 80 grand. We saved you a lot of money with this very cheap test, and you didn't have to pay. And this girl, a few years from now, 10, 15 years from now, would develop a second cancer as an effect of radiation. She would have a lot of consequences for adjuvant therapy, chemotherapy that she didn't need. She would be a survivor of a malignant tumor, but not because she had a malignant tumor. She was a survivor because she had a benign tumor that was treated. So we saved a lot of money to the healthcare system, and we always show this story to the payers, and we say, wrong diagnosis costs a lot of money. We can get it right, and you're guys are going to save the money, which actually ends very well with insurance. I'm going to show you another case. It's a 7-year-old who presented with a headache, nausea, vomiting at Columbia. There was a big tumor in the posterior fossa, leptomeningeal metastases, underwent gross total resection with a diagnosis of medulloblastoma and was treated at NYU on CCG trial 9971 with a craniospinal radiation, and did really well after carboplatin and additional chemotherapy in continuous remission for 12 years. 12 years later, at age 19, followed up with an incidental finding on MRI and new nodule enhancing in the same space where the medulloblastoma used to be. They performed the biopsy, and this is the recurrent tumor. So what is the diagnosis? Well, it looks pretty small and blue cellish, right? It's negative for GFAP, except for a few things in the background. There seems to be some synaptophysin, but is it the tumor? Is it the background? Who knows, right? Positive INI1, we always do it just to exclude ATRTs, even if it's not the right clinical history. You can see K27 was not positive, so obviously not the IPG will be unusual on this, and extremely high Ki67. So the preliminary diagnosis based on histology, not wrongly, was it's probably recurrent medulloblastoma after 12 years. A bit of a long of a recurrence, but not without the realm of possibility. But we know better by now, so when we did DNA methylation, it actually classified as a glioblastoma. It was one of the subtypes of glioblastoma that we now know is very closely associated with radiation-induced damage. It's actually a secondary GBM because of the radiation. You can see unmethylated GMT and very busy copy number profile here on the right, and you can see here subclassification. So again, if we sign out based on immunohistochemistry and histology, we'd have gotten it completely wrong. We did the NOE Lango genome pack, confirmed there was a PDGFR alpha mutations and a couple of other things that all confirmed that it's in fact a glioblastoma and not a medulloblastoma. So what are the consequences? We have a correct diagnosis. It's a horrible diagnosis. There's no doubt about it, but at least it's the right diagnosis so we can treat patient correctly. It's a GBM. And the patient was treated as a GBM. There's an epidemiological question. What fraction of recurrent medulloblastomas are actually secondary GBMs? And we never sign out a recurrent medulloblastoma without methylation because we've seen so many misdiagnosed recurrent medulloblastomas coming from different institutions. That's way too much of a medical legal problem. There is a question about survival analysis and assessment of therapeutic efficacy for design of clinical, future clinical trials. The question, how do we de-escalate primary tumor treatment to decrease the risk of a secondary tumors? That's something that we're working a lot in, epidemiologically. If most of the recurrent medulloblastomas, a significant fraction of them are actually not recurrent medulloblastomas, but radiation-induced GBMs, there's a way to rethink the therapeutic narrative. So while you may be like, Mattia, you're picking up cases that show how well it works. That's kind of like one-off, right? Pathologists get it right all the time, right? Well, these misdiagnoses have an incredible impact on clinical trials. So this was a COG trial. We were not involved with this study. I'm not on this paper. It was for children between ages 3 and 22 with a diagnosis of a PNET pineoblastoma that were randomly assigned to receive carboplatin during the irradiation. And that trial failed. And it was stopped because it was a failure trial. But what is important was all the tumors were reanalyzed using DNA methylation. 71% of children on that trial were misdiagnosed. That's not a joke. It costs $230,000 to enroll a patient on a clinical trial. Do you know how much money was wasted on this clinical trial and how many lives were wasted? Because you tell the parents that the chance of getting their child on the wrong clinical trial is over 70%. How well would that go? Right? And it also shows us we cannot be designing one bad clinical trial after another and not seeing any success if we don't rethink how we enroll patients in the clinical trials, right? So it just shows you from the beginning the trial was destined to fail. What is interesting is when they analyzed the data of this clinical trial, they showed if you only included the patients who by DNA methylation were supposed to be included in this trial, the trial would be a success. So the wrong misdiagnosis failed the clinical trial, wasted the resources, cost tons of kids their potential future, and failed. How frequent this actually happens? My current fellow, Dr. Galbraith, who is actually coming to UW to work with you guys very soon, which I think it's going to be great for you and for her, is because you guys are a great institution, has asked this same question. We are picking up hard to diagnose cases. Previous studies were all done on challenging consults. But what about in the normal clinical practice? Like, what should we use for DNA methylation, right? How often does it happen? Previous studies are all they have a selection bias, consult bias, they're retrospective, and most importantly, there are no clinical guidelines. We got asked all the time, who do you test? When do you test? How do you test, right? So she performed a study that was relatively recently published, Neuro-Oncology Advances, and we styled it as a prospective biomarker study with more than 1,900 primary CNS tumors diagnosed between 2014 and 2022, where we basically profiled every patient that had enough tumor for DNA methylation prospectively. No matter what the diagnosis was, we profiled everybody to have absolutely no bias towards the diagnosis and cherry picking what is a bad tumor, what's a good tumor. So you can hear this in panel A, you see our workflow of this clinical study, and you can see in the panel B that of all the cases we have in our study, there was about 83% that received the WHO diagnosis, and about 17%, you can see here, that have a descriptive diagnosis, a glioneuronal tumor, low-grade glial tumor, malignant tumor of unknown potential, and so on. So the number of cases that have a descriptive diagnosis is actually pretty high. And you can see that after we do DNA methylation, and in the panel C are all the cases that had WHO diagnosis, you can see that when the pathologist gives a WHO diagnosis, that means that they think they're right. In 14%, that's that blue sliver of a pie, in 14% they're actually wrong. 14% diagnostic error is something that we should all be concerned about it, right? And you can see in about 7% there is additional prognostic information. There's about 5% of cases we cannot resolve, and about 74% of cases where DNA methylation and histopathology agree. But in panel D, you see something that's very important, is how many descriptive tumors can be actually resolved with DNA methylation. And you can see that more than 85% of cases can be resolved with DNA methylation, and only 14% of descriptive cases cannot be resolved, right? And there's a couple of no-match cases. Usually it's because of a low tumor cell content, poor DNA quality. We also know that some unknown or novel drivers can contribute to a difficult molecular classification, and of course metastases. So we are now currently, I'm not going to be showing you now, we also develop a classifier for metastases for the cancer of unknown primary to be able to detect tumors that don't match on brain tumor classifier, and we want to detect where they're coming from. I always say if you cannot diagnose a tumor with 10 immunohistochemical slides, you should not go to number 11, you should just do methylation. As a part of this paper, we proposed what we called NYU guidelines for use of DNA methylation in clinical practice, which was based on this prospective study. And you can see we stratified tumors into 3 categories: high yield, intermediate, and low yield. And high yield, just to give you examples, are all the tumors that in WHO are defined by DNA methylation signatures. Every tumor that has a descriptive diagnosis should be run by DNA methylation. Tumor entities where there's a high chance of a diagnostic error, and I'm not going to bore you with it, it's all described in the paper. And then tumors where there's inconclusive or contradictory immunohistochemical or molecular results. Intermediate yield are really, DNA methylation is a very good screening test to decide whether you're going to go big DNA NGS panel, big fusion sequencing panel, what are you going to do next, and you don't want to waste too much time and tissue, so it's a good screening tool. Tumors with moderate chance of diagnostic error, and as I said, if you cannot make a diagnosis with 10 immunohistochemical slides, you should really consider doing DNA methylation instead. I love when people send me 4 trays of slides full of immunohistochemistry that's non-contributory, and they're like, oh, can you do DNA methylation? And I was like, well, there's nothing in the block, so we're done, right? And then
questions during your talk?
I don't know what you guys do. Do you guys do it usually at the end of a talk, or?
Usually at the end.
It's up to you.
It's up to you. I'm just a guest.
Okay. What do you find out if you apply that to tumors of unknown origin?
That work is in progress. I would say we can resolve about 3/4. 3/4. But I'm going to be honest with you, we sort of hijack the process. We're not trying to get it exactly to, is it a lobular ductal carcinoma, lobular ductal, is it a this or that? We're trying to tell what is the organ of origin. So we actually use developmental methylation sites that are very conserved during the development. So we just can tell, and oncologists actually, that's all they want. They say, this is what's coming from breast, it's coming from pancreas, it's coming from the liver. So it's a little bit of simplified. We don't have a classifier for every cancer in the human body, but we use these developmental methylation signatures. And then there are definitely low-yield tumors, like schwannomas, pituitary adenomas. For now, we don't have a good utilization for them for methylation, but that we think it's going to change as well with some of our prognostic biomarkers. Billing, a big one. You guys are the first one to hear that. As of last Friday, we can say that the brain tumor methylation classifier has received a specific MAAA code. And for those of you who are CPT coding buffs, you know how MAAA codes are important because they're very well paid. So labs love them, and they're incredibly hard to get. And we finally got it. It took about 2 years. So there is finally, as of Friday, we were free to say that AMA approved MAAA specific code for brain tumor methylation classifier. There's still a lot of work to do with the pricing and crosswalk and all of that, but it's there, and hopefully it's going to get a coding book as of January 2025. So it's going to be a huge advantage to those who do not have philanthropic resources like we did for the first 2 years. But I also want to point out the timeline. As I was putting this talk together, I want to show you the timeline, what it takes to take something from the research idea all the way to the clinical, right? So that's the timeline. We started in 2014. Capra et al. Published the paper in 2018 Nature. In 2019, NYU was the first CLIA lab that went live with the methylation in the US. WHO 2021 incorporated methylation in that edition, and in the CPT code was live in 2024, and it's going to be in a coding book in 2025, right? So it's about 10 years from a research to actually to clinical application. So as I always say, it takes science, devotion, and a lot of time to change the world, but it's totally worth it when you see going something from the idea all the way to the clinical application. I'm going to switch gears now we classify the tumors, we have a great diagnosis. We need drugs, right? We pathologists are great in diagnosing things and saying what everything is, but how are we going to cure anything if we don't make any new drugs, right? But with brain, there's a problem. How do we get drugs in the brain? What do we do with it? Like, there's so many drugs, but how to get them there, right? So that brings together some of my passions, which is like looking at the tumors under histology and just see how they make different patterns. And this is something that you guys are probably all familiar with, even if you're not a neuropathologist. It's a medulloblastoma, the most common malignant brain tumor of children, posterior fossa, many different subtypes. And one of them that I was always kind of obsessed with was a Sonic hedgehog. It's a cool character, always was, and a great name for a tumor and for the gene. You can see those nodules of more mature neuropil in the background, right? And then are surrounded by primitive cells, right? And that's associated with a good prognosis in medulloblastoma. Now, for those of you who are in the adrenal world, you know that neuroblastoma has something similar. There are areas of primitive neuroblasts and more mature ganglionic cells. And there's other tumors that spontaneously mature into these more like benign cells, if you will. And this was always of interest to us. And this is just an example with spatial transcriptomics. You can see in red our proliferating Ki67 positive cells, and what is dark are mature nodules in between that do not proliferate. So we've seen this a few years ago. Many other people have seen it and described it, that mature nodules don't proliferate. They look more like neurons. Immature proliferate. We did, it was in the days before spatial transcriptomics, with some laser capture microdissection, and we saw there's actually a rather small number of genes that developmentally regulates this maturation, and they seem to be regulated. You can see in GSCA analysis, H3K4 trimethyl, and K27 trimethyl mark, and then SUSY12 targets, which are epigenetic signatures. So we're like, this looks like a PRC2 controlled event, and we looked into it, and at that time I met Praveen Raju, who actually developed a model, a Sonic hedgehog patch model, floxed, that actually was showing this, this nodularity as well. You can see in about 25% of cases, you can see here on the left, were tumors that show this spontaneous maturation, and about 75% of cases were not. But what was important, as you can see in the Kaplan-Meier curve, that the ones that were blue actually lived longer than the ones that were primitive. So we had a mouse, we had a model, and we're like, wow, this actually, we can actually model this, and we can see what happens to it. We observed the same, that cells that were mature were positive for NEU-N, while the primitive cells express K27 trimethyl mark and EZH2, suggesting that the primitive cells have a PRC2 complex blockade that stops the proliferation. And I don't want to bore you with all the functional stuff that we did, but we developed an EZH2 knockout model to decrease activity of EZH2, and then we created a mouse with a constitutively activated EZH2 to overactivate EZH2, and you can see here in these bar plots that CDKNA2A and CDKNA2B are really the mechanism through which EZH2 activates. You can see that the wild type, which is the second bar, has a normal activity of EZH2, and if you knock it out, the CDKNA2 goes out, but if you do overactivate the EZH2 on the last bar, you can see that activity of CDKNA2 goes down. If you look at CDKNA2C, 1A, 1B, you don't see any change. So it really looks like PRC2 complex with EZH2 as a main actor through CDKNA2A and B are regulating maturation of these nodules. Okay, so we know there's an activity. We know EZH2 is the one who is leading it. Can we use it for something? And we can. When we have just a normal development, you can see the mature do much better than immature. But when we inhibit EZH2 through a genetic inhibition, we can actually mature these tumors and make them more neuronal looking, and we can turn basically primitive and aggressive medulloblastomas into more desmoplastic nodular, as you can see in the right side in the Kaplan-Meier curve, when the floxed purple EZH2 mice are showing significantly better outcome than the ones that are heterozygous or wild type. Okay, so we can inhibit EZH2 and we can cure the medulloblastoma, but how do we get into the brain? There are EZH2 inhibitors, but they don't get into the brain. They don't pass blood-brain barrier. It's a huge impediment of drug delivery for brain tumors, right? So how do we get them? How do we get them into the brain? So at that point, we started working on the development of clinically compatible nanoparticles that can home into the brain tumors through p-selectin on tumor vasculature. I'm going to show you that right now. So how do we make them? We use fucoidan, which is a fucosylated sulfated polysaccharide that binds p-selectin. It's incredibly safe. There's like even food supplements you can take, and some people claim there's like magical powers to make you look young. So, but for me, what matters is it binds to p-selectin, and it wraps really well into little baskets into which I can put drugs that I'm interested in. Right? Has a high affinity to p-selectin. It's very easy to build nanoparticles with, and there is more than 450 FDA-approved chemotherapeutic and other drugs that I can wrap into these nanoparticles. Right? So how does it work? So p-selectin is actually, if you guys are familiar with leukocytosis and how the leukocytes roll over the endothelium, that's where p-selectin actually really plays the major role. When there's like some sort of inflammation, it attracts the leukocytes, they roll over and they climb into the site of inflammation. And these nanoparticles that I'm showing you here, that are fucoidine based, they can also attach to p-selectin. They roll on the vasculature and they get absorbed into the tumor. So the whole idea is that because all of the brain tumors we deal with, we can't really overcome the lack for the importance of radiation. We radiate them slightly with a low dose of radiation, which induces the p-selectin expression, and then we can get our nanoparticles into endothelial covering that specifically it's in the tumor that express p-selectin. This is a really nice model, and you're like, oh, how do you guys know that actually works? And this is just an example from our recent paper in Nature Materials that you can see that if we use, if we have zero radiation, which is in the top panel, so you can see in green there is no p-selectin expression. We do quarter gray, you can see p-selectin start being expressed, and we do 2 gray of radiation, p-selectin starts being significantly overexpressed, right? In the normal brain, mouse midbrain, and panel Ds, you see no expression of p-selectin. So we can very specifically target just the radiated bed of a tumor and get the nanoparticles in there. And you see on the right side is actually a medulloblastoma from a human. We stand with p-selectin, you see the vasculature also expresses p-selectin. So we know it's really the way how we can get things in. So what we did is we took EZH2 inhibitor, we wrapped it into nanoparticles and put it in the mice. And you can see in the CDKN2A, we're actually able to get the same activity with the EZH2 inhibition as we did before. And most importantly, we can see that there is a significant extension of survival of mice when we combine the radiation with these nanoparticles in which we wrapped EZH2 into it. So, and we can replicate, as you can see here, comparison at the genetic knockout. You can see that inhibition of EZH2 on the top left panel with the inhibitor, it actually is very similar to when we knock out EZH2 genetically at the bottom panel. I showed you this before. So we're able to, with the pharmacological inhibition, get the same effect as we did with the genetic inhibition, and we can increase significantly our survival. So we did the diagnostics. We can get a diagnosis correct. We can look at the tumors, and we can develop better drugs, and I showed you how we can improve the delivery. But how do we know that the drugs actually work, right? We need a monitoring system, and it's really hard to monitor growth of brain tumors, particularly pediatric brain tumors. There's really no good tools, but I'm going to show you that there may be, and hopefully you're going to buy my argument. This is a slide that I sort of showed you before, a little bit different version, but I want you to focus on is that these 2 patients with 2 different medulloblastomas, they have also different longitudinal monitoring needs. The kid with a wind tumor, you're going to have to follow this kid for a very long time to make sure that the tumor doesn't come back. And the kid with a group 3 tumor, there's actually a pretty high chance of recurrence. And you want to spot that recurrence before the tumor metastasizes all over. So you need assays for different tumors, but you cannot be developing, like I'm sure your molecule would love to develop a different ctDNA assay for every single tumor that is out there, right? It's very practical. You want to have one assay that can do it all for all the brain tumors. So there are many challenges for liquid biopsy, and I'm specifically focusing on pediatric brain tumors because there's some unique challenges for pediatric populations. So first of all, central nervous tumors have a low number of mutations, including coding mutations. They're very low tumor mutational burden. So commercial tests that are based on the fact that you detect hotspot mutations will not work. Ependymomas don't have hotspot mutations. Many medulloblastomas don't have hotspot mutations. Gliomas may have them, but it's not a mutational burden like lung or colon, right? So you have a low mutational burden. Some brain tumors don't have any known mutations that you can target, right? And then brain tumors are highly heterogeneous. They have more than 100 plus distinct molecular subclasses I've shown you. There's presumed low cell-free DNA burden in peripheral blood due to blood-brain barrier, and I don't think that's true, but it's like a good excuse why we should not be doing it. And then brain tumors are rare. Pediatric brain tumors are extremely rare. So build any molecular assay that's going to be able to catch them is going to be really hard, right? And then in small kids, and it's something I'm particularly passionate about, is, you know, when you send for a commercial liquid biopsy lab, you send 2 tubes with 10 mils of blood. You cannot do that with an infant. There's no kid that you can pour 20 mils of blood in and send it and do it all the time. A lot of people talk about CSF. CSF is not invasive. You cannot be poking a needle in a child's back like every month. That's not normal, right? And the one thing I want to emphasize it is, because they're rare, it's really important to have a collaborative consortium type of effort to establish the values of biomarkers. So a few years ago, we started thinking about it a bit more broadly, and we were thinking, okay, what is the advantage of doing whole genome versus targeted and exome-based panel? And I would invite you to think about it this way. If you draw blood of a cancer patient, you always have 3 questions. First of all, is there any cell-free tumor DNA in the blood? Any. Did you get it? Because, you know, tumors can be small. There's a lot of blood. Is there any cell-free DNA? Is that fragment that is in your tube actually mutated? Are there any mutations in those fragments? And most importantly, are those mutations the mutations that your panel can detect? And when you do the math in your head, it becomes, wow, you got to be really lucky to find a mutated fragment in the tube of blood, and enough of them so you can actually sequence deep enough. That's why those commercial assays are 60, 80, 150,000 x deep to be able to find these mutations, right? But if you go with a whole genome, your chances significantly improve, because at that time, you're not looking just at coding or hotspot parts of the genome, you're looking at everything. Coding or non-coding, you can derive it. And you can see in this, looks like a complicated panel, but what it shows you here on the left is that if you have a single mutation panel on the very left, you have to have, it's very simple. If you have a single mutation that you're looking for, your tumor mutational burden, like the amount of cell-free DNA in blood, has to be really high, right? So the tumor fraction has to be very high. You're not going to be able to detect it. When you have 10 mutations that you're looking for, you have significantly less cell-free DNA. Logically, bigger panel, you have less, right? But if you go for a whole genome, 10,000 plus mutations, your sensitivity can go down to 10 on minus 5, which it equals what we're trying to get. You want to get the same sensitivity as BCR-ABL, right? That's the sensitivity I'm looking for, right? So the simple thing is, more mutations you're looking at, less cell-free DNA you need, because instead of going really deep, you're going really wide, right? So a few years ago, we started working on this with a company that originated from Cornell, and we built this very simple model in which we do tumor, we do whole genome sequencing at 40x, and the germline on 20x. We develop a patient-specific genomic pattern using AI, building a very specific mutational pattern, and then we follow up with a cell-free DNA sequencing on a whole genome at 20x, and we have an AI pattern recognition algorithm that takes all the mutations and copy numbers and everything into picture to be able to identify the tumor, right? So using the signature, it's a patient-informed assay, we can develop these very sensitive recognition algorithms to which we can see whether the patient is positive or not positive. I'm going to switch, so it might look weird, but I'm going to switch to lung cancer, because that was sort of like a good proof of principle for us. We went specifically for stage 1A lung cancer, which is, you know, the lowest stage, the lowest mutational burden that you can have. And patients are usually just treated with resection. However, about 20% of them recur. So it's a big problem, right? You don't want to overtreat patients with the stage 1A lung cancer, but, you know, 20% of them will recur and they will not do particularly well, right? Adjuvant therapy is not recommended. They have extremely low cell-free DNA burden. So we collected 42 stage 1A lung cancers. We derived this whole genome sequencing, derived personalized signatures, and then we followed them for up to 10 years, right? Obviously, we didn't follow them at that point. We had 10 years worth of plasma samples that we went back to in the freezer, right? So that was a fortunate thing. And you can see we had 12 recurrences and 30 non-recurrences. And you can see 2 things here. One of them is, if you're positive on a liquid biopsy, you have a significantly higher chance of clinical recurrence. But this is really what I want you to look at. What this shows is that the lead time between cell-free DNA positivity and MRI progression, right, is 16.7 months. We are able to predict a recurrence of these people 1.5 years earlier before they recur radiologically. And that's what's the key. You can predict recurrence way before radiology can predict it from the blood test, right? And this was in stage 1A lung cancers. This paper is now in submission. But we did the same thing with the brain tumors, right? So it was just to convince you that this approach is very powerful. We did a mishmash of different brain tumors, from medulloblastomas to GBM ependymomas. Every possible tumor you can imagine. We used the same principle, tumor normal sequencing, derive personalized signatures, and then look at the follow-ups in a plasma. And then wanted to see whether we can use it for brain tumors because of all the issues that I've told you, that it's really hard to follow brain tumors with any existing methodologies. And we use this, we call it AI-based error suppression model, so we can really quantify very precisely levels of cell-free DNA. I just want to show you one example that's great that highlights everything that I told you about this approach and this technology. It's like a 6-year-old patient with a medulloblastoma group 4. We specifically chose group 4 because it doesn't have hotspot mutations that you can actually track by anything else. We did a whole genome methylation. We confirmed it. It was a status post-growth solar section. There was no tumor left in the posterior fossa, and had a persistent leptomeningeal disease, and the CSF cytology and CSF ctDNA done at another institution was negative. What I want to point your attention to is, look at the single nucleotide variants, indels and CNVs, and you can see in the light pink how few coding mutations are there, right? In total, there's about 30 variants that are in the coding part of the genome across the entire genome, 30. When you look at the non-coding things, you can see it's 100-fold higher, approximately, right? You have 1,500 SNVs in the non-coding compared to the coding, 800 indels compared to 3 indels, and the copy number is 300 and something compared to 11. So the amount of information in the non-coding part of DNA is significantly higher, and that's the information that you use. It's not coding. You don't care about it for the driver, but you care about it for deriving the signature. It's much higher. So we have 2,400 mutations that we can use for this AI algorithm to recognize the signatures in the cell-free DNA. And let's cut through the chase. The first thing I want to show you is, like, see, this is a match between the MRIs and the liquid biopsy results. You can see on the left column, you can see a positive liquid biopsy from this approach, and you can see there's an arrow pointing at a very small leptomeningeal focus, right? So first of all, it shows you that this technology is incredibly sensitive, and the tumor can be rather small. That's a very small amount of tumor that we are talking about. In May 2021, you can see the tumor disappears from MRI, but we can still detect the signature in blood. And then in December, post-chemotherapy completion, you can see MRI is completely negative, but there is still a signature in the brain. So this patient recurred about 3 months ago. And we were able to predict that this patient never went into a complete remission, and although MRI was completely clean, there was always a persistent disease in the blood, and we knew this tumor was going to come back. Okay? So this shows you that there is enough cell-free DNA to monitor brain tumor patients, including the patients that don't have a lot of mutations, and is way more sensitive than MRI. Right? So it shows you the power of whole genome sequencing on the cell-free DNA. So in conclusion, I hope I showed you that molecular pathology of precision diagnostics and in-depth analysis will allow us to perform much more accurate stratification of cancer patients for clinical management, diagnosis, therapeutic discovery, and monitoring. I believe there is a significant diagnostic inter-observer variability, which is very high if it's based on histology alone. It leads to discordant diagnosis, and it can be resolved by DNA methylation profiling and machine learning classifiers. They're not ever, they don't solve everything. You need to have a human supervision to every AI, but they're very helpful. DNA methylation is the only pan-CNS tumor assay that incorporates detection of other biomarkers, decreasing the cost, turnaround time, and tissue wasted associated with the sequential molecular testing and diagnosis. It's a diagnostic tool that improves diagnostic accuracy, decreases inconclusive diagnosis, and provides prognostic subclassification. I showed you proposed guidelines for unbiased prospective cohort recommending stratification of CNS tumors based on diagnostic yield and possible diagnostic errors. I showed you hopefully the morphology, which is something we pathologists are really strong at, provides clues for further spatial transcriptomics and therapeutic target discovery. And then I showed you the maturation can be induced in medulloblastoma using EZH2 inhibition. I believe in other cancers, something similar can be done in sarcomas or other primitive tumors that you can, again, unblock the differentiation mechanism. Drug delivery across the blood-brain barrier can be facilitated using fucoidan-based nanoparticles that bind to p-selectin, and there are other technologies as well that can be used for wrapping existing drugs and delivering them into tumors more effectively. And then as the last thing I showed you, where I see the future of whole genome sequencing, It was really the huge benefit of it because it provides ultra-sensitive approach for minimal residual disease monitoring of solid tumors, including pediatric brain tumors with very low tumor mutational burden, right? So I see it as going from accurate diagnostics to therapeutic discovery all the way to minimal residual disease monitoring, combining these different modalities. And I think this is all what I have to say. I have a large team of people that is working on many different projects that I've showed you, and of course, our funding sources between the generous support of philanthropy foundations and NIH grants that enable this group. I'd like to thank you for inviting me to help to change the world here with you guys. Happy to collaborate with all of you in the future. And I was told you have to complete the seminar evaluation form, or I'm the last person who will ever give talk here. So there's a threat to all of you, and thank you very much. Pleasure. I'll be happy to take any questions.
So, yeah. And there may be a good number of people on the Zoom.
I see the Zoom. I don't see any questions in the chat, but I'm not sure if people on Zoom usually ask live or they type.
Yeah,
only do in the chat.
Okay. Okay.
In our select audience here.
I'll ask a
Other
question,
questions?
technical
Yeah.
question.
Hey.
I'm interested on just the question of sensitivity.
Mhm.
You mentioned going to the tail end of your talk, but and then I wanted to circle back on methylation as well. When you're doing the genome sequencing
Mhm.
to try to detect from a great idea to be able to be doing it from plasma, how deeply are you sequencing, or do you not need to because you have the power of multiple mutations that you're aggregating together?
Yeah, so that's a great question. One of the reasons why I thought it was very appealing to do it this way is because if you go with a target panel, your depth of sequencing has to be insane.
Right.
And it's 100,000x. What we do is we do tumor on 40x because FFP is a little more degraded and want to have a higher quality, and we do blood at 20x. At the follow-ups, you can do plasma anywhere between 20 and 30x. 20x is usually sufficient if you have a good material. I would say it helps to have a higher tumor percentage, which in brain is usually not that big of a problem. With lung, we saw 20% of a tumor cell content is usually good enough. If you go lower than 20%, then because of the FFP artifacts and whole genome amplifying all the artifacts, it becomes a little bit less sensitive. So, yeah, that's approximately what we say sensitivity. Yeah.
And then for when you are doing the methylation array, so is it about that tumor cellularity? Do you press forward? How well does the classifier work, or does it just give you basically a cannot classify if you don't have enough tumor cellularity?
Yeah, it's a good question. I mean, DNA methylation has one weakness, and that is it's very sensitive to tumor cell content because the normal cells really dilute the epigenetic signature. It was very lucky there was neuropathologists who started with it because we don't have a lot of normal cells. We don't have fibroblasts, we don't have immune cells, we don't deal with any of that, right? Have we started with lung, it would be a whole different issue, right? So brain tumors are relatively easy. Now that being said is after 10 years and all the machine learning and AI, algorithms are much better in deconvoluting inflammatory signatures from the normal. I would say tumors that have a very unique methylation signatures like IDH mutants, you can go significantly lower. Tumors that are less specific, that you really need a very high tumor percentage to be able to discern them. I would say it's always safe to be above 6, and that's true for NGS as well. More tumor cells you have, the better it is, but DNA methylation is uniquely sensitive because methylation of lymphocytes, endothelial cells, fibroblasts, whatever, it's really going to dilute the signature of cancer cells. Yeah. I see there's a question from the chat, maybe just alter. My question is, does your institution test every CNS tumor by methylation profiling? Is there any type you would recommend methylation not recommend methylation profiling? So I have a privilege of having a very strong philanthropic support for DNA methylation, and we test everything. We want to be the leaders in DNA methylation. That is our strategic initiative. We profile everything. I would say there's probably no good reason to profile schwannomas. We still do it because we want to know for some of the therapeutic things and the research things we are interested in. Pituitary adenomas, probably no good reason to using them. But I have a very talented fellow who is very persistent, and she showed me that actually DNA methylation may be able to predict recurrence in pituitary adenomas, and some of the very interesting things how expression of transcription factors does not match the DNA methylation. So some very interesting basic research things we can do. So I would say From the clinical perspective, please look into our NYU guidelines. I think that gives you a very practical approach, what should be tested, what should not be tested. In a research sense, we do everything, but from the clinical and the reimbursement perspective, those guidelines are probably going to stick around what is a high yield, intermediate yield, and low yield tumors. And we have it in the paper in the supplements, what tumors are not subject to errors, usually should not be profiled. Please, you're going to.
Yeah, to answer your question. So in your practice, you methylate everything. A lot of places do use methylation, sort of like the arbiter of whatever discrepancy, whatever diagnostic challenge there is. But can you talk a little bit about your practice and how do you resolve fairly significant differences or discrepancies between the methylation result and the histology?
Mm-hmm.
You know, you have a much larger denominator.
Yes, so one thing I want to point out to everybody is there is a huge problem if you only profile things that you know you don't know. I run 2 largest clinical trials for brain tumors in the country, and they are both for glioblastoma. And you would think that every neuropathologist, every anatomic pathologist should be able to diagnose glioblastoma. We have a significant number of errors that come through us that we are able to exclude. And it ranges from metastatic melanoma to sarcomas, to metastatic breast carcinomas, to ependymomas and everything. So I would really like to put together a study showing what percentage of misdiagnosed cases will completely destroy your trial. You do power analysis today. So if you profile only things that you don't know, you're going to be biased. And that was a problem of many of those early studies. But as Mark Twain said, it's not the things that you don't know that will get you. It's the things that you know for sure, but you're wrong, right? That's really what gets you. If you're convinced that you know all those peanuts that were submitted for that COG trial, those people knew what they were diagnosing, but they were wrong, right? So that's the advantage of being unbiased and just profile everything, what can have potential consequences. Because if you look at Kristen's paper, the error rate on GBM is about 10 to 15%. And that's a relatively simple histological diagnosis for most of the academic institutions, but it's not everybody has that much experience. When it comes to pediatric tumors, we don't take any chances. We just don't take it. The recurrent medulloblastomas, we would never take a chance because we've seen so many misdiagnosed tumors that just right now when we look at it, we think it's going to be a medical legal thing. If you have methylation available and you misdiagnose a kid and kid went on a wrong chemotherapy, wrong radiation, it's going to very soon catch up with people in a medical legal way. So we profile everything that it's intermediate yield, high yield, or we have a research questions. That's how we triage it. We have in a protocol that if methylation and histology disagree, we have to do another molecular test to resolve the disagreement. I would say in 98% of cases, methylation is correct. There's a handful of cases methylation is not correct, and it's usually because there's some unusual or weird driver. So for example, when we saw, I think I was mentioning to somebody is, we saw, for example, a glioma in a gentleman who had a germline BRCA mutation that he didn't know about. Germline mutations in unusual clinical scenarios can really throw off your classifier. But there's always an explanation, but we're always required by our SOP, by New York State, because we were so early on, they were like, okay, how do you resolve? Who's right, methylation or histology? So we have in our SOP, every discrepancy has to be resolved by a third method, independent. Yes, please.
This may be a naive question.
No such thing.
But how do you differentiate cases where theoretically a GBM acquires the methylation profile of a metastatic melanoma versus it being an incorrect diagnosis of GBM when it actually is a metastatic melanoma? You get my point. You're asking what is truth?
Yeah. So
right now the methylation profile is truth, and you're calibrating the histology to that. So how do you think about that?
Yeah, if I take you a little bit back in time, it's like how do we actually were very naive when we started working on this in 2013, 2014. We thought we were going to be done in 2 years. We're going to take every brain tumor that we know. We're going to build a reference data set of what we know. We're going to make the signatures, and then we're going to launch it. And then in 2 years, we knew this is not going to happen. So the way we started was we developed a reference cohort for every tumor that we thought was truth. For example, oligodendroglioma had to look like oligodendroglioma, had to have a 1p19q, an IDH mutation, right? Then it turned out that even when we did all this, some of the tumors were off, some of the tumors were different, right? And we started building a unique, specific DNA methylation signature for every class. Then it turned out that every tumor that we thought was one entity actually had sub-entities. So out of a sudden, there were multiple classes, and then more we did, the more classes we saw. But the beginning was always build, and that's advice I would give to everybody who's building a classifiers. First, build a framework of something that you're fairly certain about. Then you have this, we show this UMAPs and T-SNEs. You have sort of anchors of what your analysis is. And then you start profiling other things, and we start fitting them in and discovering like subsets and subsets of subsets. So first is a very good reference class. The second question you asked was very interesting is, does DNA methylation change with time and therapy? And if we do somebody who was treated, how is the classifier going to perform? So there are two answers to that. One of them is DNA methylation does change, but the diagnostic classifier doesn't care about it, because diagnostic classifier is specifically built on the signatures that remain constant over time, and they are hallmarks of that disease. So the signature, the way it works is random forest algorithm, right? There's all these diagnostic trees. I forgot how many probes we put in there. That signature will remain the same. Even if you irradiate GBM or medulloblastoma over and over, that core signature will remain. That doesn't mean the DNA methylation doesn't change. It actually changes insanely a lot, and there's a huge amount of research that us and many others are interested in. If you irradiate medulloblastoma, how is it going to look? What does it do? And everything. So, but we don't use those variables for the diagnostic. They're interesting from the research perspective, at therapeutic targeting and so on. But for the diagnosis, medulloblastoma, even after radiation, will classify as a medulloblastoma. It will show more copy number breaks because of the radiation, but it will still have those core signatures that define a medulloblastoma. Does that answer your question? Yeah, please.
I'm interested in this set of tumors that, again, that we're talking about that have a diagnostic mismatch between histology and methylation.
Yeah.
And just assuming that they were initially diagnosed on histology by competent pathologists, and
which all of us are.
Oh, exactly, right? So there's something going on with these tumors on the molecular level that's causing them to fool the pathologist on histology. Right?
Yeah.
Do these have you done any retrospective studies looking at this set? Do they behave differently than either the histologic classifier or the methylation classifier? Do they have different sets of mutation? Like, what is causing them to be tricky?
Yeah, it's interesting because when you look at a tumor, if I may rephrase your question, is tumors are heterogeneous. And glioblastoma is insanely heterogeneous, right? And I sometimes see glioblastomas with many different patterns, but they will always have that one score, right? How do you translate heterogeneity? And I trained at Mass General, and we have every Monday morning, we had a consensus conference. And that was back in times of oligoastrocytoma. So one of our neuropathologists would say, oh, this is a clear-cut oligodendroglioma. And I was like, no, it's a clear-cut astrocytoma. And the third one was like, no, it's a clear-cut oligoastrocytoma. And I was like sitting there, I was like, what are you guys talking about? Like all three highly experienced senior pathologists, everybody saw a different thing. Everybody focused on a different part, right? DNA methylation is not perfect, but it's a benchmark. It's a very simple benchmark compared to which you can do it. We do have fair amount of interest in spatial epigenetics and spatial transcriptomics, and that's something that it's one of the projects that Kristen started. Is we look at glioblastomas that have many different drivers, and we are asking, does transcriptomic changes if the cancer cells are around hypoxia, around the blood vessel, because we see histologically they look different, right? And they change epigenetically and by DNA methylation. Again, it boils down to the classifier, goes down to the single most unique denominator of that disease. And that's how the diagnostic is done. But it's an incredible amount of research that can be done on how epigenetics changes in response to hypoxia, perfusion, therapy, and so on. So yeah.
I have a general question.
Yes.
I mean, given the power, as you described it, of methylation for specificity, relates to origin, are you seeing an institutional demand at NYU for expanding your studies to all of the solid tumors?
Well, they always have to be the first guy who jumps on Normandy Beach, right? So that was the brain tumors in children. I literally had like 2-3 tumors a month. So it's pretty hard to fill those chips, right? You know how it is. But then we did all the pediatric brain tumors. Then I raised the money from a philanthropy, and I went to every hospital in the tri-state area, and I said, I'm going to profile your tumors for free under one condition. We all share the data with each other. And that's how you get people on board. That's how I filled my chips. I said, I'm going to put out the money. The only condition is we all share the data. And first people are like, oh, I'm not sharing my data with you. I want my data for myself. I was like, yeah, then you're not getting anything, right? And you convince people to share the data, you increase the volume. After pediatric brain tumors, we got all the adult brain tumors. And then people in other specialties. So now if you look at my publication record, we have methylation in lung cancers. We have methylation subtypes of triple negative breast cancers, right? We did all that because out of a sudden, everybody saw the power of this approach, right? So you just have to convince one group of people how much is going to help them, and then it just expands. And once all of our surgical pathologists believe that DNA methylation is useless to them, until there was a thoracic pathologist that called something a melanoma, which ended up being a rhabdomyosarcoma, and that was it. And you just scare one of them enough, then they realize how much of a power this classifier has. And we have Memorial Sloan Kettering uptown, right? So all our people in New York shop, right? All our cancer diagnoses go up to the big juggernaut uptown, right? So if you get it wrong, it's going to be pretty quick, right? So then people realize what a power it is that to get your diagnosis right. And the same is with the fusions, right? We have fusion sequencing, and that helps you with the diagnosis a lot, right? Same. Yeah.
Wonderful talk. One thing you haven't talked about, and let's see, I think we have about an hour before us. Louise, you're taking them up to Harvard?
Yes.
Anyway, you haven't talked about the confounders. Does aging affect methylation, the methylations and some of the other hosts?
Yeah, it does. One of my interests is, and I didn't have a chance to show you, it's a great study we're doing on the effects of race and epigenetics. It turns out that the same tumor arising in a different racial background is going to have slightly different epigenetics. Who knew, right? It's very interesting. Another thing that I'm particularly interested in is we're working on now is like, okay, if you have, let's say, African-American patient with a brain tumor, is it because of their underlying genetics, or is it because they're in Brooklyn, right? So we have a nice collaboration evolving now with a hospital in East Africa to profile some of the tumors. So I think pollution is a big one to us. Pollution really, I was in a PhD committee of a girl who was analyzing epigenetics of women with breast cancer who were in the September 11 World Trade Center because they were exposed to an immense amount of dust and pollution. And it turns out they carry those epigenetic scars with them for the rest of their life, and it contributes to the development of their cancer. So I think pollution is going to be a big one. So there's a lot of other things that co-found the DNA methylation. Again, for the classifier, because we boil down to a single simplest denominator, it doesn't affect, we think, the diagnostics so far, but there's a lot of more that we can do on further subclassifying the tumors based on the things that drove the early origin of cancer. Yeah.
Thank you again so much. This was a wonderful event, I think, for everyone who attended. So.
Thank you so much. Appreciate it. Thank you.